So good afternoon, everyone. Welcome to our PHENET webinar series addressing tools and methods for extended plant phenotyping and EnviroTyping services of European research infrastructures within the framework of the PHENET projects, EU-funded PHENET projects. We had 4 webinars in a series addressing assessment of crop and forest performance across scales, biotic interactions in agroecosystems, genotype times environment interaction across scales, and today we address the complexity of soil components. We have 5 speakers, introduction, and then Ines and Miguel will talk about building on soil ontologies. Ilya will talk about soil in ecological, agroecological settings. Then we have a group from Boku with Hans, Akos, and Pavel talking about soil properties estimation with the novel sensors. And finally, Nadia will talk about understanding soil phenology. Within the entire webinar, so related to the participants, so you are welcome to put your questions into the Q&A, but you can also most likely use the chat, put the questions into the chat. After every talk, there will be a few minutes for your questions to address them orally, or if you put them into Q&A, they could be also answered by writing. But we will address all your questions, so feel free to ask a lot of questions. Additionally, we would like to have you active, and we have a small survey. When you go to the QR code, you get to a survey where we try to get your interests in future services. So in PHENET, we are co-developing next generation tools and methods for services that are supposed to advance your research. And here we would like to specifically have your specific feedback. What questions are you interested in? What would be important for your research? And those services will be provided within the framework of this research infrastructures with ANAE addressing biochemical, biogeochemical cycles, ELTER long-term environmental monitoring, Elixir with data management, and emphasis with plant phenotyping. I will put also a link into the chat, so you can also either use the QR code or use the link that will be later on in the chat, and hope that you will be able to address the questions. It takes no longer than a couple of minutes. Normally at this point, Bertrand Muller, he's the coordinator of the project, did a short presentation. Unfortunately, he's not available today. So I would try to give you a short introduction. So I'm responsible for the work package 1 in PHENET, and would like to give you a short overview what we do in PHENET. So the reasoning why we have PHENET, so we have obviously big challenges related to climate change. There is drought issues, problems with biodiversity, for example, disappearing insects. We have extreme events, we have the soil degradation, we have human-made problems with pesticide and you name it, which results in food insecurity, health damage, and endangered agricultural systems. What we need, a transition towards agroecology-based future agroecosystems, and for that purpose, so what we need to do is biotic interactions, stimulate biotic interactions in these agroecological systems, innovative practices, use future genotypes. For that purpose, we need to understand the genotype, environment, management, species interactions to a large extent with models. And here we also need a lot of data, and the data in the end to optimize cycle duration, intercropping composition, cultivar mixture composition, stress sensitivity, which needs to be implemented across Europe as not only a European experimental platform, but this should be directly implemented to transfer our agricultural systems into agroecological systems. And here, PHENET provides the platform to address some of those goals with a number of partners and many different approaches, from remote sensing, high throughput phenotyping, field networks, instrumented ecosystems, long-term observation, data services. We have a number of EU consortia involved there, so that we provide within PHENET information about phenotypes, so basically data about phenotypes, EnviroTypes, genotypes, and specifically develop tools and methods that are part of the research infrastructures like ANAE, EMPhASIS, ELTER, Elixir. An example, what do these research infrastructures do? So, for example, EMPhASIS. So EMPhASIS addresses a wide range of phenotyping under controlled conditions, under intensive field, lean phenotyping. So pretty simple, straightforward phenotyping field sites where you can address plants. So basically understand plant environment interaction, but also evaluate this interaction. And the backbone of EMPhASIS, those are national research infrastructures, so funded by national funding, and building the backbone for basically all those facilities that jointly try to integrate their activities and provide services. There are still a lot of bottlenecks related to phenotyping and EnviroTyping. So there is a limited number of sites not covering all agroecological systems and environments that might be needed. It's difficult to translate what we do under controlled environment into a field, in particular also into a future genotype environment interactions. Data are largely underutilized, and there is a need to improve that. They are insufficiently connected, poorly interfaced, and this is where we fit in with PHENET by developing, on the one hand, low-cost, low-energy Internet of Things-based networks of sensors, developing high-resolution Earth observation services, but also driving data and models. So basically making data fair, and in the end also explore digital twins, artificial intelligence, developing models for predictive modeling. In more detail, we are organized in PHENET in 4 technical work packages addressing the devices with embedded AI, so basically a number of sensors that are developed. Then we have Earth observation services, which is more or less remote sensing applications that are used and developed there. Then there is open science, basically data management work packages that addresses all these digital services, and simulation intelligence services addressing modeling, for example, digital twins, hybrid modeling. Those technical work packages contribute to use cases. So we have a set of use cases, in total 9 of them. They are grouped into biotic interactions in agroecosystems, where we look at plant health, bumblebees as in bioindicators, but also intercropic settings. We have genotype environment interactions on the one hand in orchards, specifically in apples, but also in cereals. We have a complexity of soil components. That would be the topic of today's webinar, where we have soil phenology, but also soil health. And we had also one topic assessing crop and forest across scales, where we look at the regional development in a dollar valley, but also farm to form to platforms where we specifically address use of remote sensing and research infrastructure tools to specifically address the needs of farmers. The way how we approach the project is practically this development. On the one hand, the work packages and the use cases is this kind of a matrix where we have this four technical work packages. They provide development and tools to the specific use cases, and they develop the technology based on specific scientific questions within the use cases. And this is complemented also by work package 6 and 7, where we have outreach, innovation, and training based on the tools that we are developing. As such, this is the entire group from the kickoff meeting, I think. And as such, we can move on then to the next presentation. So we will have 4 presentations today. And as mentioned already, please write your questions into the QA or the chat, and don't forget the survey. We would really appreciate if you could fill in the survey. It is very valuable for us and hopefully also for you at some point, where we have services that are adapted to your specific needs. And as such, I would then invite Miguel to the first presentation about building soil ontologies.